Good afternoon, everyone. I'm Janani Gnanavelan. I'm from Physics Department. Today, I'm going to present my research work titled Stochastic Modeling for Semi-Discontinuous DNA Replication in T4 Bacteriophage. I'm working with Dr. Deepak Bhatt, Department of Physics, Vellore Institute of Technology, Vellore. So we all know DNA is the molecule of heredity. For a cell to divide, it must first replicate its genome. In other words, the entire DNA has to be precisely copied before segregation. Understanding how this copying occurs at the molecular level is one of the central questions in biology. And it's here a physics-based model will offer some insight. So we know DNA has anti-parallel structure. One strand runs from 3' to 5', other runs from 5' to 3'. There are a bunch of proteins involved in the process, collectively called as replisome complex. One of the key protein is called DNA polymerase, which can able to synthesize the new DNA only in the direction of 5' to 3'. For the template strand, 3' to 5', it is straightforward for polymerase to synthesize continuously. That continuously synthesized strand is called So here, the template orientation has opposite direction, which causes the DNA polymerase to synthesize in backward direction. Both strands are synthesized simultaneously. To maintain the coordination, the polymerase on lagging strand synthesizes short fragments. And these short fragments are termed as Okazaki fragment. So this continuously synthesized strand termed as leading strand, and this discontinuously synthesized strand is called lagging strand. So this process is called semi-discontinuous DNA replication. So using this simple picture, I'll introduce a few core replisome protein in T4 bacteriophage. One is helicase, which is used to unbind the DNA. Another one is polyprimase, which used to synthesize a short segment called primer, which is then extended by DNA polymerase as DNA. The clamp loader is used to load this DNA polymerase specifically at this priming location. Additionally, there are several other proteins like single-stranded binding protein, clamp, ligase. All this will help for Okazaki fragment maturation. So even though the DNA replication process is well studied, but several dynamics of proteins are still unclear. Especially here, the dynamics of DNA polymerase, how it unbinds, all these are unclear. So we are focusing on the lagging strand polymerase. So in this process, consider priming occurs stochastically. So polymerase extend the DNA upon encounter the previous fragment, or it can unbind before encountering the previous fragment. With this picture in mind, I'll introduce our model and assumptions. So these are the main assumptions we consider in our model. There are only two polymerases in the replication. One is leading strand polymerase, another one is lagging strand polymerase. These polymerases attach to the replication fork, and we assume both of these polymerase moves with the same speed v. So we consider the lagging strand polymerase bind randomly with rate pi, and it can unbind in two possible ways. Either it can unbind randomly with rate epsilon, we call that as a spontaneous dissociation, and it can unbind by hitting the previous fragment. This process often referred as collision in the literature. So here the gap appears only when polymerase unbind by collision. Gap occurs only when polymerase unbinds spontaneously. If you see polymerase unbind due to collision, there is no gaps between the Okazaki fragment. So these are the information are taken from the experimental studies of bacteriophage. And so we are building the model based on the experimental evidence. And so for this talk, I'll show results first. I'll keep the technical details at the end. So in this model, we also consider these priming, clamp loading in terms of binding rate. And the special collision with previous fragment can be considered in collision. The other molecular interaction which cause polymerase to unbind before colliding can be considered in terms of spontaneous dissociation. To understand this more simply, I have two limits. One is spontaneous limit, another one is collision limit. So if you see epsilon tends to infinity, tells you polymerase will unbind immediately. So it will unbind, so naturally it won't hit the previous fragment, it unbind immediately before hitting it. That is spontaneous limit. If you take epsilon tends to 0, it will take long time to unbind. So naturally, it will hit the previous fragment and dissociate. So these are the two limits in our model. We will study exclusively these two limits in our model. So let me show the Okazaki fragment size distribution and gap size distribution, because we are interested in these size distribution, which will reveal the underlying polymerase dynamics involved in lagging strand synthesis. So let me show the expression for Okazaki fragment size distribution. Q of Z, Z is the fragment size. I write A of Z in terms of Q and R. So Q is epsilon by V, R is pi by V. So Q relates to unbinding, R relates to binding. So we extracted the experimentally reported Okazaki fragment sizes from T4 bacteriophage system, and we identified the binding rate and unbinding rate. So they have reported for two different primase concentration. One is 8 nanomolar, another one is 64 nanomolar. So previously explained, we are considering priming, clamp loading in terms of binding rate. Here you can see for 8 nanomolar, a low primase concentration, a binding rate is smaller than for the high primase concentration. We also found the average Okazaki fragment size, which is found by 1 minus Fc by Q. So here Fc is the fraction of collision. I explained polymerase can be unbind due to two mechanisms. One is spontaneous collision and spontaneous dissociation. Another one is collision. So here Fc denotes the fraction of collision. So here these symbols are taken from the experimental data. The solid line represents theory. So similarly, we identified the binding rate and unbinding rate of the polymerase. We fixed the V also, which is 400 base pair per second, taken from this literature. So similarly, we will predict the gap size distribution now. So R of G is the gap size distribution. I referred gap meaning the single stranded space between two Okazaki fragments as a gap. So you see there are two parts. One has contribution from collision and other has contribution from spontaneous dissociation. I previously told, like, whenever polymerase unbinded due to collision, we could not see gap between the fragment. So that can be represented by delta of G. So here we fix the pi star and epsilon star by previous fitting. So we plot gap size distribution for the same parameter. Here that insert shows the peak of the distribution, which is governed by this delta of G. So it kind of telling that majority of the time polymerase dissociated due to collision maybe. So these are the average gap size we found, which is 1 by R. If you average over only the non-zero gap sizes, which is mainly contributed from spontaneous collision, can be written like 1 by R into 1 minus Fc. Fc means fraction of collision. So you can see from here also for 8 nanomolar means small R value, 64 means large R value. For large R value, the peak shifted to the lower gap sizes. So we wanted to study more about this fraction of collision and how it contributes to the system. So we derived the fraction of collision in terms of Q and R. So when we plot for different R by Q value, we can see by increasing R by Q value, the fraction of collision will increase. These two are the experimentally reported condition for 8 nanomolar primase and 64 nanomolar primase concentration. It is majority of the time, mostly more than 75%, the polymerase dissociated due to collision. So we will also check by plotting this with respect to average Okazaki fragment sizes. For increasing R by Q value, we can see the contrast behavior, which is the average Okazaki fragment size reduces by increasing R by Q value. So we also see in terms of size distribution. Here I plotted Okazaki fragment size distribution for different R by Q value. The green, red dot from these figures have these distribution. So these three dots I have plotted the distribution. For R by Q equal to 1, meaning the fraction of collision is very less in that case, the Okazaki fragment size distribution has monotonic nature. This is completely different from what we observe experimentally. So then you see we slowly increase the R by Q value, which has more collision. The size distribution looks non-monotonic. So this in a way suggested that the collision indeed plays a role in the polymerase dynamics. So I summarize all these results in the following table. So for high binding rate, we observed a smaller average Okazaki fragment size and smaller gap size compared to the low binding rate. which tells because the fraction of collision is higher in the high binding rate compared to the low binding rate. So now I show the outline of how we derive all these size distribution. So we start by writing the Chapman-Kolmogorov equation, like probability to synthesize n plus 1th Okazaki fragment of size g and gap size g. We can return like this. So Z prime and G prime are the previous fragment size and previous gap size. And this W, Z comma G from Z prime, G prime tells you the transitional probability from previous gap size and fragment size to current fragment size Z comma G. So we'll proceed with this following picture, and this is how we approach this. So we apply steady state condition to this equation, then we marginalize with respect to dg and dz. So we will get separated distribution for q of z and r of g. So we solve these equations by using Laplace transform method. And that's how we derive all the size distribution and Okazaki fragment distribution. Thank you, everyone.
Thank you. Questions? I have a question. So in the eukaryotes, there is a study which said that the Okazaki fragment length distribution depends on the nucleosome length distribution. So equivalently, I am wondering in bacteria, are there some protein distribution that also regulates the
So
Okazaki?
1 second.
So in eukaryotes,
Okay.
there was a study which argued that experimentally they showed that the Okazaki fragment length distribution, which is what
Okay.
you have been
Yeah.
computing,
Yeah.
It depends on the position of nucleosomes. Like 150 base pair is
Okay.
200 base pair is the distance between two nucleosomes. So it had a peak at 200 base pair, and they said that this falling off, which you call the collision, which happens at the next nucleosome.
Oh, okay.
But I am wondering, is there anything equivalent in bacteria where you have some other protein sitting there and it hits the protein and then the collision happens? I'm just curious, but do you, if you're aware, you can...
I'll think about it, yeah.
You had spontaneous
Unbinding.
hello? Yeah. You have spontaneous unbinding on the lagging strand.
Yeah.
But did you have a spontaneous unbinding on the leading strand? I don't know why I don't understand why there's a difference.
In leading strand, polymerase can able to synthesize continuously. It no need to unbind frequently. So it is the behavior of lagging strand polymerase because it was moving in backward direction. It has to unbind frequently and attach to the entire replication complex. That's why we consider spontaneous unbinding on lagging strand only.
So it's the same DNA polymer that's...
Yeah.
Thank you.
Could you explain to me what is the sort of implication of a collision-based method for determining the Okazaki fragment size as opposed to the gap-based method that you had previously?
1 second.
What is the sort of implication of such frequent collisions for Okazaki fragment determination? So is there a cost associated with such frequent collision-based unbinding and then rebinding in terms of efficiency of replication and so on? Have you calculated such things?
No, so in this model, we are mainly focusing which plays major role in the polymerase dissociation because continuously in literature, people are telling it's not due to collision, it's due to some other protein interaction, like the primase synthesis gives signal to polymerase, which dissociate before hitting the fragment, all these phenomena. So we wanted to test whether which plays a role. So we consider only 2 cases here. One is collision and another without collision with previous fragment, whatever comes can be considered in terms of spontaneous dissociation. And I showed 2 limits in the model. So we check for a spontaneous limit, we can able to observe the monotonic size distribution, which is not experimentally measured. The experimentally measured distribution is always non-monotonic nature. So we kind of ignored. So the spontaneous limit does not play any role actually. So then we try to understand more about collision, and we found that collision indeed plays in the average fragment size, reducing it or increasing. So we can predict based on if you use this binding rate, if polymerase bind use this binding rate, we can able to see a larger fragment size or a smaller gap size. So we can use those numbers.
Okay, then no more questions. Thank you, Janani.
Thank you.